So you might think that compared to determining DNA structure and everything, it should be easy to determine protein structures the same way. Well, the first protein structures appeared just a few years after the DNA structures, but that was a tour de force work by a number of groups, in particular around the Laboratory of Molecular Biology in Cambridge. So one of the first structures to be determined was this one. It's hemoglobin, and it's the molecule used to carry oxygen in our blood. Max Perutz is the hero behind this one, and it took them 22 years to discover it. I can't even imagine. Today it's easy because we know that they have a unique structure that we can determine, but of course Perutz didn't know that. So that they spent 22 years going after and cracking that problem, which I think is an amazing scientific achievement and probably a sign of some astronomical stubbornness. Here's a picture of Max, and can you see that insanely large model there? Because again, what we do in computers in 2 seconds, they would have to do manually and carefully, measuring with a ruler, and then you have these rods going bottom up so that you can push each atom and place the atom in a slightly different position, and then measure again and see, does that correspond to the expected diffraction pattern that we would see in the experiment. Although this was done without computers, it's very much the type of modeling I spoke about. You come up with a model, you test your model, you should be careful not to fall in love with your model, and then you assess your model against the toughest experimental results you can find. And of course, when you're successful, you tend to change science, such as Max did here. Today, we're blessed enough that we have many more proteins with known structure than when I was your age. I think Sture Forsén in Lund, for whom I took biophysical chemistry in the mid-1990s, early 1990s even, he had to strike out the line in the lecture compendium that there were like 30 known protein structures, and said that there's almost 300 now, an amazing amount of them. Well, today there are well over 100,000. And since the day I took this, we even have structures of membrane proteins. That might not sound remarkable to you, but the first structures of membrane proteins was bacteriorhodopsin. And that appeared a few years before I was a PhD student. KCSA ion channels, 1998. That was while I was a PhD student. The first ever ion channel structure determined. This is the corresponding human protein. There's actually a trend here. It's kind of conceptually the same channel, but a prokaryotic protein is somewhat smaller and the eukaryotic one is somewhat larger. We're going to come back to that later on. They've both been love stories in different parts of my life. This is another membrane protein, aquaporin, which is the channel that is regulating water contents in and out of cells. There are actually a bunch of aquaporins that are present in your eyes and others. I won't have time to go through them all. But super important discoveries. And Rod McKinnon and Peter Ager, they shared the 2003 Nobel Prize for Chemistry for these discoveries. The GPCRs that I already mentioned, Brian Kobilka shared the Nobel Prize with his advisor for this in 2012, and Ray Stevens has also done very important contributions to pharmaceutical design of this. And it might sound stupid that we're awarding so many Nobel Prizes of these protein structures, but it's literally before we have these structures, it's like a black box. We know that they're doing something, but we have no idea why or how. The second we have these structures, even when they're somewhat low resolution, we can usually describe the entire molecular process and understanding, aha, it's binding something up here. That is changing the protein to another stage, which is releasing the signal here on the other side. And that in turn is then used to, say, design drugs and alter this behavior of a particular protein. So make no mistake, I think that we're going to keep seeing Nobel Prizes awarded in structural biology.